from Stanford University, and they will be speaking about active sink particles, how sessile filter feeders affect sinking aggregates.
Great. It's so nice to be here. Shilpa, thank you for that introduction and for the group for organizing this. It's really nice to see some of you, even though it's virtual. Good to see everyone. I'm excited to talk about some work that Deepak and I have been doing. As Shilpa said, I'm Rachel Pepper. I'm at the University of Puget Sound. And Deepak is here somewhere and I worked with him and Manu at Stanford. And this collaboration actually started at a conference where I heard Deepak talk about this gravity machine that he and Manu had invented, which you'll hear more about later in the talk. And I thought this is a really exciting opportunity for a collaboration. And then we actually fleshed out the collaboration and really made plans at the Ocean Biophysics Whistler meeting. So this group and the meetings that we have has really been important to getting the work done that Deepak and I are gonna talk to you about. And Deepak says his computer crashed, but he's about to rejoin us. So he'll be back, he'll be here shortly. And so we're doing this work on active sinking aggregates, and we actually did the work. We were lucky enough to do the work at the Hopkins Marine Station in California, and this is a view from right outside the lab. So it's a terrible hardship to be at this beautiful location doing our work. And I've been thinking about these active sinking particles because I think, as most of you know, aggregates are a really important part of aquatic ecosystems. For instance, marine snow, shown here, are a really critical part of the ocean. And many of you, I think, know about marine snow. In fact, more than me. I'm a relative newcomer to the field, and some of you are real experts. But for those of you who are less familiar, marine snow are these really abundant aggregates in the ocean. And I love this quote from Rachel Carson that sort of sets the scene for them. The steady, unremitting downward drift of material from above, flake upon flake, layer upon layer, a drift that has continued for hundreds of millions of years, the most stupendous snowfall the Earth has ever seen. And these abundant particles are biological debris. They're sort of fluffy and amorphous. They're small. They sink at intermediate Reynolds number. And they're really important because they're responsible for most of the vertical fluxes in the ocean. And so understanding things like their sinking rates, aggregation rates, size spectrum are really important for understanding nutrient budgets in the ocean, or things like carbon sequestration. So really almost all ocean processes. And not only are they important, these sinking aggregates important in the ocean, they're similarly important in freshwater systems, in estuaries, and even in wastewater treatment plants. And in all of these places, they're also biological hotspots where they're densely colonized by bacteria and larger organisms like flagellates and ciliates. And one class of these organisms that they're colonized them densely are these microscopic sessile suspension feeders. And what makes them unique is that they're sessile, they live attached to the aggregate, and because they don't swim, they generate a feeding current to bring their food to them. So here's an example of a Vorticella, one of the larger sessile ciliates that attaches, and its ciliary motion and the feeding current it generates. And so there's a large range of sizes and activities of these filter feeding organisms that can be attached to the aggregates. And we started thinking about how the flow of these attached organisms could combine with the sinking flow in interesting ways. And to give just another example of these active sinking particles that have flow generating organisms on them, I thought I'd show you the Tacoma wastewater treatment plant. So this is pretty close to my house in Tacoma. And wastewater treatment is actually one of the really critical processes in modern life that we don't think about all that much, but it's really important to having clean water. And the final stage of cleaning our water for many treatment plants that use the activated sludge process is these settlement tanks where the treated waste has formed flocks like the one you see here. These flocks are made mostly bacterial biofilm, and they have attached things, ciliates here, you see the Vorticella. And the final stage of the cleaning is the sinking out of these flocks that have waste and organisms on them. And that's what cleans the water. So the sinking aggregates, again, play a critical role in the wastewater treatment. And let's see if I can successfully turn this over to Deepak to give one more example.
Yeah, sorry about that. My computer crashed just as the meeting was starting. Okay, so let's see if I can do this. Yeah, if not, Rachel, you can just
Yeah, I
advance
can
the
just advance it
slides.
for you.
Mhm. Yeah, and I think as Rachel mentioned, there are many examples of these aggregates which are associated with these sessile suspension feeders. And we got a very nice glimpse at this during one of our field trips where we could see these Vorticella attached to these diatom cells. And more recently, there's been more work, again, looking at a similar kind of system where you have the centric diatom with these Vorticella attached to it. And just to kind of give you a glimpse at what this looks like, we were able to measure the flow field around that particular aggregate that I just showed you. And you can clearly see this, the flow due to the sinking of that aggregate, but also this significant modification of the flow due to the filter feeding current that you have here. And so this is going to kind of set the stage and give you a glimpse at what we are going to be looking at today, which is to really think about these as active sinking particles. And in terms of, kind of placing them in the context of what people have thought about before, I mean, you can really think of them as lying in the spectrum, where on the one hand, you have a passive sinking particle which doesn't have any biological activity. And on the other end, you have an active swimmer where activity dominates its near field hydrodynamics as well as motility. So active sinking particles lie somewhere in the middle, and you can parameterize that as a ratio of active and buoyant stresses. So active sinking particles are going to lie in this middle here, where you have competing effects of both uh, sinking and activity. And the rest of this talk, we're going to really ask this question about how the effect of this activity, what effect this has on both the flow around aggregates as well as the mass transport. And what we are hoping to kind of show you is that we have a new experimental system where we can make these kinds of multi-scale measurements for the first time. So I want to give you a quick outline for the rest of the talk. And yeah, of course, the motivation behind that is understanding vertical material fluxes in these ecosystems. And so I'll briefly talk about the experimental system that allows us to make these measurements. And then I'll start by telling you about the flows that we can measure around these active sinking particles. And then I'll turn it over to Rachel again to talk about the mass transport. Cool. With that, we can dive in. And to actually make this measurement was an interesting challenge because as many of you might know, these sinking aggregates in the ocean, for example, can sink at rates between 10 to 100 meters per day, right? But at the same time, we are interested in these microscale processes that are happening, such as the flow field around these aggregates. And as we just saw, these can be controlled by single cells. So how do you actually bridge those scales between the microscale and the scale of hundreds of meters? And this was something that we scratched our head a lot about and actually came up with a simple idea where we can actually make such multi-scale measurements. And some of you may have seen this work presented in the Whistler meeting. So we call this scale-free vertical tracking microscopy. And the idea is very simple. So we use a circular fluidic chamber, and we use the fact that for very small objects, we can actually compensate for movements along the vertical direction of the circular fluidic stage. So what you essentially have is a hydrodynamic treadmill. And what we can do with this technology, which is kind of like developed now to the stage where it's actually a 3D tracking microscope. So not only can we measure these vertical displacements, but we can also track the object in the other two directions. And this is a glimpse of this microscope in action, actually at Hopkins Marine Station, where, as Rachel mentioned, we collected most of the data that we will show today. And that's actually Rachel collecting some of the data you'll see. So yeah, if you're interested, do take a look at this website. And I also want to say that this technique in this particular talk, we are going to mainly use it to look at sinking objects, but it's more general. So you can actually use it to study behavior of, active biological swimmers. So here is an example of a villager larva. So this is the larva of a snail. And you can see this beautiful helical swimming trajectory that it does as it swims through the water column. So yeah, if you're interested in knowing more about this technique, we have a website, so do take a look. So for the purposes of this, of measuring active sinking particles, what we can do with this microscopy is to combine it with another method for creating model aggregates. And here what we did is created spherical agar spheres, agar beads, where we can actually control the density of this sphere, and therefore actually tune the sinking rate. And we can incubate them with Vorticella to essentially have our model active sinking particles. So you have an agar sphere with attached Vorticella. And these sink at rates of around 1 millimeter per second, and the Reynolds number is around 1. So I want to give you an example of what the data sets look like. So here on the left, you have the scale of the entire trajectory, which can be around a meter or a few meters. For example, this track was around 0.7 meters in 30 minutes. But at the same time, you can also measure local hydrodynamics. So you can measure the flow around the aggregate, and you can also resolve flows at the scale of a single cell that is sitting on the aggregate. So you can see that here. So that's a single Vorticella creating feeding currents sitting on the aggregate. So with this kind of multi-scale data set, we can actually do some interesting measurements, which I'll show you next. But as a baseline, we wanted to establish the flow around like a passive sinking particle, right? So here, what I'm showing you is a freely sinking aggregate. So many of you might have seen these kinds of flow visualizations before, but remember that here the aggregate is not really tethered or fixed in the lab reference frame. It's actually freely sinking, and we are tracking it using this technique. And we have added tracer particles to the fluid so that we can actually visualize the fluid flow structures. So in this case, what I'm showing you are the fluid path lines using this technique called flow trace, where we look at the path lines over a 1 second interval. And you can see this kind of expected flow field for a passive sinking particle. And this serves as a very nice baseline to compare and contrast what happens when you add even a few Vorticella. So you're going to see that in the next slide, where, so if this movie plays for everyone, you can see that there are actually two Vorticella kind of sitting on this aggregate here, one at the top and one near the bottom off to the left side. And you can see that the flow fundamentally looks different from that of the passive sinking particle. And on the right, you have a more dramatic example where the Vorticella are, again, there are two of them that you can see, and their orientation basically means that the flow looks completely different from that of passive aggregate. So even from this very simple measurement, you can see something that many of you might have expected, which is that the flow is qualitatively different, and it's also sensitive to both the location and the orientation of the Vorticella. So with that, we have one more very nice movie to show you. And you can kind of imagine that as you increase the number of Vorticella, you get more complex flows. And you can now see that the near field flow structures near this aggregate look nothing like the passive sinking particle. So you can already anticipate the effects that this would have on things like mass transport. And to kind of look at that more quantitatively, Rachel will kind of tell you about how to quantify these flow fields and what we can work with them.
Great, thank you, Deepak. So because we had the tracer particles in there, we could do particle image velocimetry and actually measure the flow fields around these freely sinking particles. So here's an example of that for one of the aggregates that didn't have any Vorticella on it. And I kind of like this one because it has a few kind of protrusions that make it not completely spherical. And you can see both in the flow trace streamlines and in the particle image velocimetry, the expected flow field around a spherical sinking aggregate. And then we could also measure the flow fields around, here are those two example videos that Deepak showed you again, but this some still images where we've done the particle image velocimetry around them and measure the flow fields. And again, you can really see clearly that the number of Vorticella, their locations and their orientations really determine how much that flow field has changed from the more subtle changes in this case to the more dramatic changes where we're getting those recirculating structures. And we're showing you sort of 3 examples, representative examples out of a set of videos that we took. And overall, we were able to do particle image velocimetry where all of the action was sort of in the plane of focus, the direction of the Vorticella, and also the central plane of the sphere for 12 different flow fields and 7 unique particles. So some of the particles, we had multiple Vorticella orientations on there. So I will show you all 12 of those in some aggregated data later. So with these flow fields, we were able to think more quantitatively about things like how the near field flows and boundary layers are changed. And the way we did that was by thinking about taking a transect of the flow in this ring around the sphere. It's about 100 microns away, and that just allows us to think more quantitatively about how the flow is being changed. And what we did was looked at the perpendicular flow and the parallel flow, so perpendicular and parallel to this line here around the sinking sphere. And here it is for this one that has no Vorticella on it. And you can see it's pretty smooth. There are some disruptions around these protrusions here and here. And the dashed line is what we would expect for a Stokes flow, so 0 Reynolds number flow around a sinking sphere. And you can see that this sphere, even though it's at lowish Reynolds number, something a little less than 1, it's matching quite well the Stokes flow with a few deviations where the protrusions are. But the story is quite different when we add the Vorticella. So again, they're here at the top and at the bottom left. And so you can see that in the in this graph here and here are where the Vorticella are. And what you can see is even in this relatively subtle modification to the flow, it actually is pretty dramatic when you look at it quantitatively. So the flow is slowed down on one side of the Vorticella and sped up relative to what you expect. The radial flow is greatly increased at both locations of the Vorticella. And so you can imagine, even though this is pretty subtle, it is fairly dramatically changing the velocity boundary layer, and probably also then the mass transport. Areas that are faster will have a higher mass flux, and areas that are slower may have a lower mass flux. And then in this case where the Vorticella are causing recirculation, you can see that the boundary layer is totally reshaped on the side that has the Vorticella. Faster and slower flow slows the opposite direction that you would expect. You have this whole area of stagnation here that you wouldn't have in a bare sphere. And again, you can imagine that this would dramatically alter mass transport to the sphere in ways that depend both on the orientation of the organisms, but also on where you are along the sphere, whether you're close to an organism or not, whether you're in an area of stagnation or area where the velocity has been enhanced. So I think it's pretty clear that these near field flows are dramatically changed in a way that's likely to change mass transport. And we've also used our flow field measurements to think a little bit more about how these particles and their mass transport may interact with the ecosystem and environment around them. And we've done that looking at sort of 2 properties of these sinking aggregates. One is the plume they've left behind, and the other is the encounter volume that they encounter as they're falling through the water column. And both of these are ways that they interact with the water column around them. So the plume, as shown here in a study done by Thomas Kirbo and collaborators, is an area of either enhanced or depleted solutes that form behind the sphere as it's sinking. For instance, we could think of this as leaking amino acids, and so this area would be enhanced in those leaking amino acids. And it's really important to understand the plume because this is the signature that the sphere leaves behind, and bacteria and copepods can follow this signal, either chemotax to the aggregate to either colonize it or consume it, depending on the organism. And the other sort of ecosystem level interaction that we look at is the interaction volume below the sphere. So this is work by Stuart Humphreys, and here it's sideways. So the sphere is sinking from right to left or flow from left to right. And so as it sinks, depending on the particles it might be encountering, there's a volume of fluid that anything within that volume of fluid will actually directly touch the particle as it sinks. And that's an important concept to think about if we're thinking about aggregation or flocculation rates, because that volume of fluid and what's in it will determine the growth rates and dissolution rates of these sinking aggregates as they fall through the water column. So there's a couple ways to think about these plumes and these encounter volumes. One of them, like Thomas and his collaborators did, was to use the theoretical models and fully solve the advection diffusion equation to get a concentration field here. And that's a little bit beyond the scope of what we want to do, because we want to focus on what we can get from these cool experiments that we've done. Although now that we have the experiments, they'll can be a great jumping off point to validate models and do this in the future. But what we did was something following more the method of Stewart-Humphreys, which is just simply follow the streamlines in our measured flow. Because we have a measured flow, we can get streamlines. And neglecting the diffusion and simply following streamlines is a pretty good first estimate for sinking aggregates because they have a very high Peckley number, which means effective transport is dominant over diffusive transport. And actually, because of that, the near-field flows of sinking aggregates has long been recognized as a really important in controlling their mass transport. So another indication that the changes in near-field flows that we see could be really important for changing mass transport. So this Peckley number, the ratio of effective transport to diffusive transport, if we think about the plume case first, it could be molecules like oxygen or small amino acids. If we put their diffusion constant in to find the Peckley number, we find Peckley numbers on the order of 1,000 to 10,000. So you can see much larger than one, so effectively dominated. And then advection is even more dominant in the case of this encounter volume, because what we're encountering are sort of large, say 100 micron other aggregates, or maybe non-motile bacteria that cause the aggregate to grow in time. So they have really small diffusion constants and very large Peckley numbers on the order of 10 to the 7th or 10 to the 8th. So again, advection really dominates. And another way to look at this that may be even more appropriate than the Peckley number for sinking aggregates is to think about the Sherwood number that doesn't think about rates, but thinks about total mass transport. So this is the total mass transport to the sinking aggregate over the diffusive mass transport, and it's a common dimensionless number for both sinking aggregates and swimmers. So we're able to use the work of Thomas and his colleagues to look at this in the range of Peckley numbers and Reynolds numbers that our sinking aggregates have, and also the range that is appropriate for marine snow. And we find that the Sherwood number is around 5 to 10 for bare spheres in our range. So again, the mass transport for things like the plume and the encounter region are dominated by advection. So as a first estimate, we just follow streamlines to think about them. And the way we do that is we start with a ring around the aggregate, again, about 100 microns away. And all of these points that you can, I don't know if you can see them, little blue rings, we start a fluid particle and then just evolve it in our measured flow field. And that's what these purple lines are. And we go backward and forward in time so that we get both above and below to get the plume up here and the encounter region down here. And so the width of this plume is highlighted with the yellow lines, and that's our experimental measurement. And I've also compared that to a couple different ways of calculating it theoretically, given the sinking speeds, the Reynolds number of our particles. So the solid orange line here is what we'd expect for Stokes flow, so 0 Reynolds number. And then if we add in intermediate Reynolds number and duocene flow, which is appropriate for low Reynolds numbers, that's the dashed line. So you can see here, again, it's the same 3 particles we've been using as examples so far. You can see that for this bare aggregate, we have quite a smooth plume and encounter volume, very similar to what we expect from simple theoretical calculations. However, if we look at the case with Vorticella attached, in both cases, the more the flow field that wasn't disturbed as highly, and also the one with the recirculation, we can see that both the encounter volume and the plume are dramatically changed. In this case, the plume is much thinner. The encounter volume is now not axisymmetric anymore, possibly thicker. I can't quite tell. Over here, it's really reshaped, and the encounter volume is much larger because of the orientation of this Vorticella right here. It's really pulling in things from much farther away than this aggregate would normally encounter. I do want to point out that these are 2D slices of a 3D flow field, so it may be that this plume is thinner in this dimension and thicker in the other. And also the Vorticella move in time on a time scale of minutes or so. So these will also be dynamic in time as the Vorticella change their orientation. And we're able to quantify the width of this plume and encounter cross section. So this is just the distance between the 2 yellow lines here. So here, this is the encounter region, so it's the region below. This axis is the vertical distance from the center of the sphere. So here going downwards, this is that width up here. And I scaled everything by particle radius just so we can more easily compare these 3 that have different initial radii. And what you can see is here, the 2 cases with the Vorticella on there have a thicker encounter region than no Vorticella. And in the plume, we sort of see the opposite. As we move away from the sphere, everything gets smaller, but the cases with the Vorticella have thinner plumes that they're leaving behind as their signature. And one last thing we were able to do with this, thinking about the encounter region and the plume, is really for interacting with their environment. It's not the plume right here in the region that we can measure directly with our particle image velocimetry that maybe determines their encounter with the environment, but the plume further away. So we took our measurements and actually used theory to start from our measured widths and evolve them further away. So here we took as pretty much as far away we could get in our field of view, about 2 radii away. We then took those widths that were experimentally measured and used a theoretical ocean flow to evolve those streamlines from 2 radii away to 20 radii away. And here are those combined results for all of our measured flow fields now, all 12 of them, although not all of them did we have the right field of view. So I think there's actually 10. So here on this axis is now the number of Vorticella, and this is that width, that long distance width for the plume. It's up here at 20A. And shaded in gray is a theoretical prediction, what we predict for a bare sphere at the Reynolds numbers that we have. And what you can see is that our plume widths for attached organisms are both larger and smaller than what you would get for a bare sphere. And we see the same thing with that long distance encounter region, the region below the volume of fluid that the sphere will intersect as it's falling. Again, we see both larger and smaller. And so what we think is that the attached organisms create a very different plume and encounter region to a bare sphere. The way the plume and encounter region are different is very sensitive to the Vorticella location and orientation. It will be both thinner and thicker, maybe thinner in one dimension, thicker in the other, maybe dynamic in time as the Vorticella change their orientation. And this could really have implications for how easy or hard it is for organisms to colonize the bacteria, and also for the growth and degradation rates of these particles as they're thinking through the column, if they're able to encounter more or less, and if that's dynamic in time. And this theme of dynamic in time actually was bigger than we initially thought. And I'll let Deepak pick up there and tell you a little bit more about that.
Yeah, I think one of the cool things about doing experiments is you see things that you don't expect. And one of the things when we were doing these experiments, we noticed is that not only are the Vorticella changing the flow around these aggregates, but they were doing something more than that. So here, what we found is that these Vorticella can actually, cause these aggregates to spin around and rotate in a way that bare aggregates don't. So here, I'm going to kind of show you like a quick guide to the eye. So I'm going to track 2 points just on the aggregate surface, just to kind of, show you what the orientation is. And you can see here on the left, you have an aggregate with no Vorticella. And you can kind of see that it's sedimenting at like a relatively fixed orientation over time. But then when you do have Vorticella on the aggregate, you see something quite interesting. So you see the aggregate actually start to rotate. And the effect seems to get stronger with an increasing number of Vorticella. So all these movies are played over the same time duration. And you can see even with your eye that the sphere on the right rotates much faster. So we were actually kind of curious enough to look at this more quantitatively. And to do that, we came up with a pretty simple but kind of useful image processing trick where we tracked these surface features that naturally occur on these aggregates. So these can be Vorticella, these can be like debris, that's small debris that's stuck to the aggregate. And what we can do is we showed that if you can track 3 or more of those points, you can actually extract not only the rotation rate, but also the axis of rotation. So you can actually take 2D images and extract 3D rotation vectors from them. So this is something that we did on our data sets. And this let us actually quantify this active orientation changes that we saw in the previous slide. And you can see here that I'm going to make this rotation rate non-dimensional by scaling it by the rotation rate that would be due to a single Vorticella. So that's kind of like, imagine a Vorticella that's actually pointed exactly tangential to the aggregate. And since we know the average force that Vorticella exert on the fluid, we can actually rescale this rotation rate by that, and therefore also remove any possible dependence on the aggregate size. So if you do that, you still see that the dimensionless rotation rate changes, it increases with increasing number of Vorticella. And you also see that the distribution of those rotation rates, like just think of that statistical distribution, you actually see that the distribution becomes more wider as you increase the number of Vorticella. So what this is telling you is that this dynamic change in the position of the and orientation of the Vorticella is kind of like self-fulfilling. So it's just the activity automatically changes their orientation. And to kind of like put a picture in your head, this is kind of like a satellite where there is no other forces. So even tiny rockets on the satellite are going to be able to change the orientation of the satellite pretty effectively. And what this means is also that we can actually measure the rotation axis, as I said. And it's interesting to look at how the rotation axis actually changes with the number of Vorticella. And what we see is kind of interesting. So you see that the rotation axis for the case with no Vorticella is pretty random. So you see that here. So I'm plotting the orientation of the rotation vector in the azimuthal and the polar direction here. And what you see is that the no Vorticella case is pretty random, but then you start seeing rotations that are actually aligned along the axis of gravity. So this is something that we don't fully understand yet, but what we think is happening is that the aggregate has a stable orientation related to a gravitational field, and these Vorticella are actually causing precession about that stable orientation. So in any case, what this means is that for the purposes of all the things that we looked at so far, the flow field, mass transport, encounter, and plume cross sections, these are all expected to be dynamic in time due to the effect of this rotation. So with that, I want to kind of summarize what we have told you today. So we want to stress that this is actually just a starting point. So we have kind of shown that you can use this experimental system to make these multi-scale measurements of active sinking particles using this model aggregates and gravity machine. And this is really a starting point to understand how the flow around these aggregates is changed by this activity, and to really think of these aggregates as a new paradigm for looking at transport in the water column. And yeah, as Rachel already mentioned, these change both the near-field flows and the boundary layer. And as I just mentioned, the rotation rates are also actively driven by these Vorticella. So to kind of think of this as a new paradigm for thinking about sinking aggregates is, I think we should really care about how activity modifies the near field flows and therefore the mass transport to these aggregates. And it's actually very interesting to ask, like, what is the Peclet number scaling for an active sinking particle? Is it somewhere in between what you see for a passive sinking particle and that of, say, an active squirmer? And these measurements are just a starting point. And I'll let Rachel kind of tell you like the larger implications and future things that we are excited about doing with this.
Thanks, Deepak. Yeah, I think Deepak really laid that out sort of nicely, that this is maybe a new way to start thinking about these particles that we've illustrated on the left, where instead of the hydrodynamics of them being passive as they sink, the hydrodynamics are active, where they're interacting with their environment with consequences to maybe lots of different things. And our measurements are really the first point to be able to ask questions like this. And we're hoping that these microscale and multiscale measurements that we've been able to make will be a jumping off point to think about how the microscale processes interact with some of the ecosystem level processes. For instance, we're hoping that we can understand how these active particles change the growth and decay of things like marine snow, how they affect the nutrient environment for the organisms that colonize in marine snow, chemotaxis and colonization rates, and of course, as I think through the column, how it changes the aggregation rates, size spectrum, and thus sort of sedimentation rates and the rates of nutrient transport throughout. And more specifically, to have some next steps we're pretty excited about, sort of, since this is so dynamic, thinking of good ways to find average mass transport rates of sinking particles, as Deepak mentioned. Being able, the gravity machine is a really exciting tool that I've been learning more about as we've been collaborating, and it allows us to do things like take long-term measurements of aggregate fate with and without the organisms to see if aggregation rates really are different between the two, to measure the mass transport and nutrient environment around them as the two are sinking. And we'd really like to take measurements with natural aggregates that have whole communities on them, because we've sort of done one aggregate size with one organism on it. But in reality, there's lots of different organisms of different sizes, aggregates of different sizes and properties. And so we're excited to explore that. And from a theoretical perspective, we'd like to also do a numerical solution of the advection diffusion equation to understand how mass transport and concentration fields right nearby are altered. And one of the reasons why I was excited to talk to this group is you all always have so many great ideas. So we'd love your feedback and to hear your ideas for next steps too. And with that, I'd like to thank those who made this possible, our funding sources, the Prakash Lab at Stanford, the Pepper Lab at the University of Puget Sound. I'd also like to thank Anders, Thomas, and Emily at DTU, who have really shaped our thinking on this. And I'd really like to thank you for listening. And with that, we'd be happy to take any questions or comments. Thank you again.
Thank you. And I guess we'll open the floor for questions. I saw a few come in through the chat, I think. Does someone want to start from the chat? Marco, maybe?
Yep. So thanks a lot for the talk. It's really nice. I was wondering for the rotation and sinking. Actually, there's a couple of questions if I can. So the first one, it looks like I was wondering whether your idea of the stable position due to gravity might compare, how that might compare to, for example, stable position due to the fact that these protrusions will then make an anisotropic sort of drag tensor, right? So therefore, as it sinks, it will have a specific sort of at least one stable fixed point. That's one thing. So I was wondering why you went directly to gravity. So maybe there is a reason there. And the second is the Vorticella. Do you think that they generate, in a sense, each one generates a torque on the object? And then if you consider the different Vorticella as generating sort of random torques, then as you put them together, there should be a specific scaling for the rotation versus number of Vorticella, whether you have tested that.
Rachel, do you want to take one or both of those, or?
Yeah, sure. So yeah, actually, I think the drag anisotropy is a great idea. We sort of brainstorm a lot of ideas, but I don't think we came up with that one. And so I think we'll have to think more about that, because that certainly seems as plausible as having sort of a heavy spot somewhere in the particle as well. Mm-hmm. And the scaling, Deepak, correct me if I'm wrong, but I think that's another great idea that we talked about, but we haven't done. So it would be really interesting to see if because it was increasing, but maybe not as rapidly as you might expect if you don't take random orientation into account.
Because I think that there is a paper, I think was 2019 probably, of Ray Goldstein on the rotation of aggregates of conoflagellates. I think, nice. I sort of remember that, where they were talking about the rotation rate with that aggregates or randomly oriented particles would have. So maybe, I mean, it might just be that you can apply that to this case as well.
Oh, that would be great.
I think that's a great suggestion. And yeah, we have thought a little bit about both the drag anisotropy and the rotation torques. And I think you can see that, like, the number of Vorticella that we have, I think we are still in the limit where, you know, there's not enough Vorticella for the averaging effect to start taking over. And it'll be interesting to see in these natural aggregates, which have many, many more, what happens. And I think that's why it'll be fun to actually image natural aggregates as well. Yeah, but that's a great suggestion.
Are there any other questions?
So this is Jules. I actually have a hand up. Hi.
Sorry, I missed that somehow.
No, that's okay. Just trying to be tactful, actually, which is not always one of my asses. So great talk. Love to see it. Two comments. One sort of old guy comment about we had developed a plankton treadmill many years ago, but never did any science. So congrats. It's great to see the science coming out of this. And then I just want to make a little bit of a pitch for in situ microscopy. The PNAS article that just came out, I sent a copy of a picture from our peer microscope where we see these linear diatoms with really tons of epiphytes on them. And the speculation is that those are actually Vorticella. And I would be happy to send those pictures to anybody. But I think the role of the Vorticella in infecting or populating diatoms may be less about flux and flow And it's an interesting sort of question about the ecology. And I guess there were some ideas about that in the PNS articles. So if folks are interested, I'd be happy to send them a copy of that picture. It's a fascinating topic for me to think about. So thanks again for a great talk, you guys. And thanks for listening to my questions and comments.
Thanks.
Thanks, Jules.
I see another hand up.
Yeah, I was like, Fuad, is that?
Yes. Hi. This is from Harvard School of Public Health. I'm pretty far from this. I still like the biophysics microscale. Thank you both, Rachel and Deepak, for the talk. I've actually saw Manu Prakash talk last year in Hawaii about this gravity machine, and I fall in love with it because I think this is really amazing invention. So my question is just, I know it might be hard, but could you imagine of a system that turned this machine into an incubator, a simple incubator, so you can follow cell division, a different other process, because we can look at phenomena from biophysics perspective on plates, on agar plates, but what if you transform this gravity machine into an incubator that can last at least for 24 or 48 hours, and you track all these phenomena? Do you have any thought about that?
Yeah, I can take that. So thanks for the question, Fuad. I hope I'm pronouncing your name correctly. Yeah, thanks for the kind words and the question. And yeah, I think that's a fantastic idea, and that's something that we are definitely thinking about quite a lot. So I'll definitely say that both in terms of like, thinking about cell division over ecological scales, like over 24 hours, as you mentioned, and then just using the chamber, like the circular fluidic chamber that we use for tracking as a place to grow organisms and like grow not only one organism, but more than one organism. Actually, those are all kind of active things that we are looking at. So yeah, I hope you'll see some work come out on that. And like, yeah, definitely, that's a great suggestion. Yeah. And I think we are also, as we move forward in the technical development of this tool, we are definitely including those kinds of designs into the design of the circular fluidic chamber. One of them is like, so there are, some of you may have seen, there are actually these microgravity incubators that people have developed for culturing like aggregates, aggregated cells. And so we are trying to essentially look at some of those ideas to kind of keep a constant nutrient concentration in the chamber, for example.
Amazing. And just one little thought as well, because I have more evolutionary background. So I think about that if you can, at any time, like transform this into continuous tracking, it can be transformed into evolutionary experiments, a long-term evolutionary tracking experiment.
Yeah, that's a great idea. Yeah.
Thanks. Are there any other questions? I don't see any other hands raised. Well, if not, let's thank our speakers again. We can give them a virtual applaud, and we really appreciate you guys being here. We will have another seminar in September, so look for that announcement. We are planning to have them monthly for the fall semester. And then we also wanted to let you know that the conference is planned to be in person as of now, and it will be either in May or June in Majorca. Is there anything else I should add, Marco?
No, for the moment, no. I mean, as you know, we are discussing the details, but yeah, hopefully it's going to be in presence in here. Yeah, I mean, I don't know what to add. I don't want to jinx it.
Yeah, and then I was like, we have 15 more minutes or so in the hours. If people want to hang out and chat, we can leave the room open. And
I think Rachel can stop the recording now.
Yeah, someone should stop the recording.